This is an OCR A-level biology video on biochemical tests. We're also going to be looking at colorimetry, biosensors and chromatography in this video. Last video we looked at proteins and inorganic ions. If you want to watch that you can. But let's get into this one. So let's get this title written out quicker. So this is a picture I took of my notes. And I know it's a very lazy way of doing it, but why do it twice? So, biochemical tests, it's pretty simple. You just have to learn it. It's not a matter of me really teaching much to you. Just learn each test and learn it well. Okay, so easy marks if you can know all these tests and how they work. So first of all, starch. It's the iodine test. So we add iodine solution in potassium iodide to a sample. When dissolved in potassium iodide, the iodine forms a triiodide ion which slips into the middle of the amylose helix, causing the colour change. And that colour changes from yellow-brown to blue-black. Okay, starch, iodine test, yellow-brown to blue-black. Then we have reducing sugars, so this is our Benedict's test, and we heat the reducing sugar with Benedict solution in a boiling tube in the water bath at 80 degrees for 3 minutes. Copper ions are reduced to copper 2+ is reduced to copper 1+, which forms an orange-red copper oxide as a precipitate, which is solid and in solution. This is more of a gradient that you produce, so this is why we use colorimetry later. So we go from blue to green to yellow to orange-red. So orange-red indicates the most reducing sugars. If you're getting a green or a yellow one, we're seeing less reducing sugars, but reducing sugars are still present. Okay. Then non-reducing sugars, which you should remember from our carbohydrate video. First we'll do one test for reducing sugars. If you don't get results, then we boil it in hydrochloric acid to break the bond. Then we neutralise it with sodium hydrocarbonate and we cool it down. Then we can repeat our reducing sugars test, which I've already spoken to you about, with Benedict solution, and we'll be looking for the similar results. So some substances contain both reducing and non-reducing. If they do tests equal amounts for both, and the non-reducing sugar filtered precipitate should weigh more. Okay. Now lipids, the easiest test. It's the emulsion test. Take a sample, mix it with ethanol. Any lipid in the solution will dissolve in ethanol, because we know lipids are soluble in ethanol but insoluble in water. So we dissolve them in ethanol. Pour the solution into water, and we should get a cloudy white emulsion, which indicates the presence of lipids. We call that our emulsion test. Proteins, I said lipids was the easiest one, but this is probably even easier. You literally just add the sample to a white tile, add burette reagent. The colour change is formed by a complex between the nitrogen atoms in a peptide chain and the copper ions. Effectively, the test is for peptide bonds. So pretty much we're detecting peptide bonds which are only found in proteins, and we get a colour change from light blue to lilac. So really quick overview: starch, iodine, yellow-brown to blue-black. Reducing sugar, Benedict's solution, blue to green to yellow to orange-red. Non-reducing, carry out our reducing sugars test. Hydrolyze the bond with hydrochloric acid, break it, and then neutralise that with sodium hydrocarbonate and cool it, and carry out that reducing sugars test again, and we should get more precipitate produced at the end. It should weigh more if we've got non-reducing sugars. Then we've got lipids, emulsion tests, so we dissolve them in ethanol and then add it to water, and we should get a cloudy white emulsion. And proteins, we use the burette reagent test, where we just literally just add it to the solution. We get a colour change from light blue to lilac. So they are qualitative tests. So food samples have to be mashed, grinded, mixed vigorously with water before, and you should always wear eye protection when carrying out these tests. What they do is tell us the presence of something, but they do not tell us exact quantities. They are not a quantitative test. So one way to make this a quantitative test is by use of colorimetry. So usually when we use colorimeters, we're looking at the reducing sugars test. Okay, so we're looking for that gradient, so blue to green to yellow to orange red. So we remove any precipitate by either leaving it for 24 hours so the precipitate settles out, or we can centrifuge it. So using a colorimeter with a red filter, measure the absorbance of Benedict solution remaining in each tube. You should use a negative control at first of pure water, just to make sure the curve is being calibrated correctly. You should use the results to make a calibration curve showing the absorbance against glucose concentration. The higher the Benedict solution, the more blue it is, therefore the more absorption. Okay, so if we've got lots of Benedict solution, it means it's unreacted and we've got a low glucose concentration. So low glucose results in high absorption. High glucose causes low Benedict solution, so it becomes less blue. There's lowered absorption, and therefore we get, and that is a result of our higher glucose concentration. So high glucose causes less absorption. Low glucose causes more absorption. Calibration curves are then used to work out how much exactly, or work out the concentration of glucose in our sample. Right, so now we can have a look at biosensors. So they're another way of getting a quantitative statement for how much concentration of glucose, etc. we had. Okay, so a biosensor is a device that uses biological molecules such as an enzyme to detect a chemical. So it contains a transducer to turn this chemical signal produced by the biological molecule and convert it into an electrical signal. And this electrical signal is then processed and can be used out to work out other information. So the electrical signal pretty much gives a reading of what concentration there is. An example of a biosensor is glucose biosensors. So a glucose biosensor is used to determine the concentration of glucose in the solution. It uses the enzyme glucose oxidase and electrodes, and the enzyme catalyzes the oxidation of glucose at the electrodes, creating a charge, which is converted into an electrical signal by the electrodes, the transducer. So the electrical signal is then processed to work out the initial glucose concentration. So that was biosensors. One final thing to look at is chromatography, which is a good way for separating and identifying things. So it separates things in a mixture. It then can be used to identify the components. So for example, we can use chromatography to separate amino acids, carbohydrates, vitamins and nucleic acids. There's different forms of chromatography. Electrophoresis is a form of chromatography, but the ones we're going to look at here are paper chromatography and thin layer chromatography. So in all chromatography setups, we've got a mobile phase where the molecules can move. It's usually a liquid solvent such as ethanol or water, and then we've got a stationary phase where the molecules can't move. In paper chromatography, the stationary phase is a piece of chromatography paper, but in thin layer chromatography, the stationary phase is a thin layer of solid, e.g. a silica gel, glass or plastic plate. Okay, so stationary phase, they can't move through it. Mobile phase, they can move through it. The mobile phase moves through or over the stationary phase, carrying with it the different solutes. The components in the mixture spend different amounts of time in the mobile phase and the stationary phase, and if they spend longer in the mobile phase, they travel faster and further, and usually that is the smaller molecules, the lighter molecules. The time spent in the different mobile phases separate out the molecules. So what I do here is draw on our chromatography setup. So down here we've got our solvent in a bowl basically, and we've got our chromatography paper here. I'm just colouring in our solvent. We draw on a pencil line near the bottom of the piece of chromatography paper, and a concentrated spot of the mixture. So for example, we could use amino acids, and we put a concentrated spot of these amino acids on our pencil line, and we call that point the point of origin. We do not put the solvent over this, as it will just then diffuse into our solvent here. It will not be carried up the paper, it will ruin the test. So we just put the point of origin just above the solvent, and the solvent is carried. The solvent here is our mobile phase, whereas our stationary phase is the chromatography paper itself, and our solvent moves through. So one thing I didn't mention to you is, because we're dealing with solvents, we want to be using a fume cupboard and lid for safety, and it also helps prevent the evaporation of the solvent. That's why we put a lid on top of it. You do not want to be breathing solvents. So this is what happens. The solvent is carried up the stationary phase, carrying with it the different solutes which are moved in the mobile phase. We draw at the top our distance moved by solvent, which is the solvent front, and as we can see, things have been carried up the solvent at different rates, and these are our spots. So if we're dealing with amino acids, these end up being clear, so we have to use an anhydrine solution to turn them purple. So we spray it on the amino acids, and we help detect the different amino acids and just see them on the spotting tile, and we can work out by comparing previously known data to which amino acids we've got where. Okay, you should also wear gloves when doing this experiment, and one thing you can work out now, which is a question that will often come up here, is the R1 value, which is the ratio of the distance travelled by the spot to the distance travelled by the solvent. And you can calculate it using distance travelled by spot over distance travelled by solvent. That produces your R1 value. This spot will have a lower R1 value than this spot, as it travelled much closer to the R1 value. And I'm pretty sure, I've not been told this, but I'm pretty sure your R1 value will always be less than 1, because you cannot travel further than the solvent front itself. Okay, unless it travelled exactly onto the solvent front, where you draw the solvent front and you've got a spot on the line, then you get a value of 1, but other than that you will not get a value above 1. So that is chromatography, all our biochemical tests. Just remember that these tests here are qualitative tests, whereas here colorimetry and biosensors to a certain point are quantitative tests. But chromatography is just a way of separating molecules. It's not really a biochemical test itself. Okay, but it does show you which chemicals are present. That was a really long video on something that should have been really short to cover, but thanks for watching.